So we're going to start things this morning with Dan Clare from the eBIC Centre, and he's going to give you a roundup of all the exciting developments that are happening here on the RAL campus. Dan, the floor is yours.
Big build-up. Well, I'd like to thank Tom and Martin for giving us the opportunity to give a presentation on eBIC. So eBIC is the national EM facility. Is it still a national EM facility? Well, who knows? But yeah, so this was originally from an award from the Wellcome Trust MLC BBSRC to Helen, Kai and Dave to set up a user facility with high-end equipment. So the aims was to offer state-of-the-art cryo-EM facilities for both single particle and cryotomography. It's going to be funded access based on scientific excellence to the instrument time, and it also includes some provision for containment facilities, Cat 3 at Oxford, at Struby. We're also, on our remit, is to run some training courses to try and widen the access to EM to both other structural biologists and cell biologists. The plan is also to have an in-house research group headed by a director, and to kind of link Cryo-EM with CCP-EM, EMDB, and the EU through iNext. So the user access, so that is at the moment 80% of the microscope time that we have will be given to users. We have a couple of modes of access. So rapid access was the standard, is the one that we initially rolled out, and that's 2 to 3 month kind of peer-reviewed, and it's an allocation generally of 2 days, and this is kind of load dependent. I mean, at the minute we're kind of really heavily oversubscribed, so it's been pretty difficult to give everybody the time that they would like. We're just starting to roll out the bag access, which is similar to the type of access that's offered to the MX beam lines, where you have multiple groups that submit proposals on multiple projects, and then time is allocated over a 2-year period with a kind of with progress reviewed after 6 months. The acceptance criteria, it's based on scientific excellence, so it's peer reviewed by a group of EMPIs, who probably most of them are in the audience, and then ranked based on that and time allocated. Travel's covered for UK users, so 1 to 2 people for visit, and that includes accommodation. For EU visitors, you can apply through iNext, which will also pay for your travel, and this will eventually fund UK users to use eBIC as well once we try and get remote access up and running on the machines. So the management structure at eBIC, so we have a management group which is Helen, Kai and Dave, plus Martin Walsh, who's sitting in the audience. We report to a scientific advisory committee that has at least one member of the EM community on, and the user program is run through the user office, and as most of you have seen, with proposals and things like that. The core staff, the target is to have a director, 2 senior facility scientists, 2 more junior facility scientists, that should be a PDRA, but never mind, RA, and there's a lot of technical support on site with Diamond, so a lot of engineers, vacuum detector experts and things like that. So at the minute we currently have 3 full-time staff, so me, Alistair, who is probably in the audience, and Corey, who's just started. So the equipment we're going to have, so at the moment we have, the plan was to have 3 TEMs. So we're going to have 2 Titan Cryoses, which are the latest 300 kV machines from FEI, 1 Talos, which will be used hopefully as a screening stroke project development microscope. So at the minute, Cryos 1 has a full user program running on it. Cryos 2, the installation is getting close to completion. We've had some cryo images from with an automatic run set up on the Falcon 2, but we're just waiting for the second detector to be fully installed. So all of the microscopes are equipped with the latest generation of direct electron detectors. So the 2 Cryoses both have the Falcon 2 with the upgrade voucher and the K2 Summit on the end of a Gatan energy filter. For sample preparation, we have a VitraBot, we have access to a high pressure freezer, we have a SIOS dual beam FibSem instrument, which is currently located in Oxford, and we'll have access to cryo fluorescence microscopy and super resolution cryofluorescence as well. At the moment, because we're still in the kind of startup phase, so I mean, me and Alistair kind of only started full-time at the start of the year, and as I say, Corey's only just started. We're expecting people to ship frozen grids, but I mean, in the future, when staffing levels increase, we'll be able to help out with sample preparations as well. So this is Cryos 1 on the hall floor. So this is right next to I-20, I think was on Dave's slide. But so this was put in, the installation started in May last year. It was kind of had beamed down and running pretty much by June. Some initial cryo data in June. We accepted it on a Friday and we opened for business on the Monday. So it was one of the fastest installations around, and since its opening for business, it's not stopped collecting data. So it's flat out. So we are meant to, as you can see, this is actually on the main hall floor of the synchrotron. We are meant to be moving to our new building, so that was handed over in January. As I've already said, we've got a lot of people who want time, and we can't, at the moment, give them the time that they need, but hopefully with Cryos 2 that will ease that a little bit. But because of the explosion of the popularity of EM, we've also decided that maybe 2 Cryoses might not be enough. So when the building was handed over, so this is obviously the building, you saw this from the canteen yesterday, it's just across the way. It's strategically located close to lunch, so that's good. Alistair will be happy. So this is our kind of corridor. So on the left-hand side are our main microscope rooms, and on the right-hand side we have room for the FibSem SIOS, the Talos. We're probably thinking about getting a screening microscope, a smaller machine, and then we're going to have cell culture facilities and a preparation lab for cryo. This is just a Talos room, and so this is the actual main room, which is now, so originally we had 2 6x6 meter rooms, which we've now knocked together to make one 12 by 12 meter room, and this is hopefully where we'll house many Cryoses. We'll see. So Cryos 2, that installation started at the end of January, and there's been a few issues along the way, but it's getting there. So you can see that was the frame, then they build the outside with the panels, then the microscope goes in, and then the GIF gets put on at the end because it wasn't done in the factory. So the EM data flow. So at the moment, we're running obviously the Falcon 2, which is a 17 frames per second camera, and we also have the K2, which is a 40 frames, but we generally run it at a much lower rate than that. So the data rate on the Falcon 2, generally we can get between 50 and 100 movies per hour, depending on parameters, how many frames you write, your type of grids you use. For the K2, it's a bit slower, but again, depending on what type of grids you use, you can get up to 75 images an hour, which is not bad for saying the exposure time is generally 5 to 6 times longer at least. So all of the data that's collected on the microscope is written directly to our main file system, which means as soon as the image is written, you have access to it via FTP, or Globus if you have that. And all of the data will be on spinning disk for 40 days, but after that it will be, it's archived to tape pretty much straight away, and it will be stored on tape for as long as those tapes last. And we also do have a cluster which, during usual running of the beamlines, is pretty full with MX jobs, but certainly when the beamlines are shut, there's a lot of computer power sitting there that could be used. So the typical user day, I thought I'd put this in, okay. So generally our sessions start at 9 a.m, so most users are kind of early. We tend to load generally about 8 grids, but that can vary. The maximum with the Cryos is 12. But the proviso is the more that you load, the longer it takes to do that, so the less time you potentially have to collect images. So this takes about an hour. I mean, this could be speeded up. I think once more people have maybe Taloses or Cryoses and have the auto-loading system, if you can bring already pre-clipped grids, that saves quite a lot of time. It's much faster to load into the cassette directly. So then we'll load the grids into the Cryos, generally, about 1020-ish. Then most people's grids that they bring are very good, so the screening is more trying to find the optimal grid, so the perfect grid with the good ice, with the nice particle distribution, low contamination. So that can take, I've put an hour and a half here, but sometimes that can take a fair amount of time. So then once we've found the optimal grid, we would collect a grid atlas, which you'll see an image of on the next slide. Then we would set up EPU, check the microscope alignments, do detector gain references, particularly for the K2. And then between 3 and 4, we'd probably be checking images, so looking at, is the defocus range right for your particle, things like that. Is it accurate? How far are the tone rings going out? Various things like that, which can all be done in a graphics room that we have upstairs, which has access to pretty much most of the EM software is on the Diamond system already. And then hopefully, if this is all good, we collect for 40 hours, and me and Alistair generally monitor this remotely. So the software that we're using, this is for single particle. We're also using the FEI software for tomography, so Tomo 4. But we use an EPU, so this is just for most people have seen this already. So this is your atlas, so this is your grid. These are your squares, then you zoom into your grid square, and then you can zoom into the hole. And then depending on your hole size, depending on what magnification you're using, you can set up multiple acquisitions in the same hole. You have a focusing area and a drift measurement. And then EPU, once you set this up, it will automatically go from hole to hole, collecting these images for you and show your summed high resolution image down here. This slide's from Sonia from FEI. So far, as of Monday, we've had 78 external user visits. 51 of those are from unique groups. We have 200, or if it was all on disk still, we'd have 220 terabytes of data. We're averaging just over 2 terabytes for a 48 session on the Falcon 2, and a bit more on the K2. So that's kind of equivalent with our standard parameters to about 2,500 images in 48 hours is kind of what we're running with optimal grid type. We know of multiple structures between 3 and 4. I know of at least 2 that have gone to better than 3. And so this means in the first year of operation, we will have provided 220. So that's projected, because obviously we've still got a month and a half left. But obviously we're fully booked, so I kind of know what the numbers that we're going to reach. So that will be 220 days for external users, which is 40% over what was. So I think it was a total of 200 days that we expected the microscope to be up, with 80% of those going to users, so 160 odd days. So we're doing all right from that perspective. I think it shows how stable the Cryos is, and pretty much it, so far, touch wood, it's not broken down. So, yeah. So then these are some statistics from just the number of unique groups from where they are, and effectively how much of the time has been allocated. So you can see that we've pretty much covered most of the UK's EM community. We have a few groups from Spain, we have Stockholm, and we had our first US visitor a couple of weeks ago. So, yeah, but you can also see there's a lot of Cambridge users. So when we had the first Cryos installed, we did some commissioning tests. So this was one of the first ones from Abe and Alistair and Sonia. So Sonia, again, at the start was doing a lot of the setup and data collection. I mean, at the moment, me and Alistair are kind of doing more of that. But yeah, we wouldn't have been able to do that without FEI's help at the start. So this was rotavirus, so it's just a quick test, 300 particles, 4.4 angstrom resolution on the Falcon 2. Then we did another test again on the Falcon 2 with GrowEL. I've shown these slides before, so I'm probably going to go quite quickly through them. So, you can see raw image, class average, some bits of density chopped out. So this was an average resolution of about 4.1, but it looks certainly in the equatorial domain, so that's this bit here that you've got some rigid regions that are higher, and it's a process would rely on. And again, this was Sonia set this up. And then obviously about, a couple of months into the first year of running the Cryos, we had the K2 integrated into EPU. So this was the first attempt at integrating the Gatan filter plus detector into FEI's data collection software. So we had a few teething troubles initially, but since then it's been very good, very good. So, and this was, literally the first run that we tested was with everybody's favourite GrowEL. I was trying to persuade Neil to use that on their Cryos, but he didn't seem too happy about that, so he's just bitter, that's all. So then we've got images again, class average, you can see some density. So this was, a 10-hour data collection, 18,000 particles, 3.3 angstrom resolution. So I think it's just been published as part of a methods paper that Agnel did in Maya's group at Birkbeck. And you can see again the equatorial regions are very nice. I'm not sure it's really 3.3 angstroms in the apical domain. So within the first 6 months we had 2 papers published. So you saw Dorian's very nice talk yesterday. You'll see Gabriel's very nice talk this morning. So yeah, it's going very well. It's going very well. So the next set of slides I'm going to show are kind of work in progress. So this is something that we would like to roll out the user program, but we're still trying to optimize the kind of workflows to make sure that this works consistently for people when you're collecting automated data, and that's a bit tricky. So, I mean, everybody is interested in the faceplate. It's going to effectively give you contrast for free. So it basically shifts the phase of the unscattered electrons such that it converts them into a kind of an amplitude contrast, so you don't have to, so you can work at focus and still see your object. So this should allow you to collect in focus, artifact-free data, high resolution single particle. When I first put up this slide, I think 4.4. We're now, I think the recent paper from Danev's group is now at 3.2. So I think that's proven. So you just have to be very, very careful with the way that you collect the data. I think this is the one that's most important for the faceplate. It should allow access to much, much smaller proteins because of the massive increase you get at the lower spatial frequencies. So the kind of frequencies that are represented by the size of your object will be much, much more than you would be used to. So it just makes it much easier to see your object. So this was from the CWAT when Alistair was out at Eindhoven. So our Talos will also have a faceplate on. So this is in focus, and this is without the faceplate and with the faceplate, and the difference is pretty spectacular. The other thing is, this is really going to help with tomography, where you're really working at the edge of signal to noise. So any kind of contrast increase you can get will be really, really beneficial. So recently, with an in-house session, we were trying with kind of a test object. So this is from Dave's group. So this is from Abe, Liz, and from Dave's group. So this is FMDV. So it's about a 30 nanometer virus. So if you look, this is normal TEM at about 1 micron under focus, and you can see them. They look very nice. But this is now the same sample, effectively, with the faceplate in at focus. So I think most people can see the difference. So it's pretty amazing, to be quite honest with you. And then if you look at the power spectra, so the real benefit from using the faceplate is you don't have these irritating CTF zeros. You don't have to correct for that. So the problem is that you have to make sure that you're at focus or very close to focus, and that's the trick. So I went away and just did a quick analysis of this because I think Abe is going to do the full analysis. So this is these were 5-second exposures, 25 frames. So I've just taken the sum that EPU writes out, so no movie frame alignment. So this is accumulated over the full 5 seconds. I've picked out the micrographs that were what I deem close to focus. So if I go back to that previous slide, so if you see here, so this is the diffuse water ring at about 3.7. So what I'm going on is I can't see any zeros to 3.7, so I'm assuming that I'm close enough to focus that I shouldn't be affected by the CTF. We collected. The issue we have at the moment with EPU is that we can't control regeneration of faceplates. So, albeit it's automatically collected, it means I have to stop the run every hour or every hour and a half and recondition a new faceplate. But that will be fixed certainly in the next version of the software, and I think can be done if you have serial EM and some macros is I think what Danav uses. From 8,500 particles from about, I think we collected a couple of hundred micrographs. I've used about 130 of them, 8,500, 8 hours. We've got about 5.2 angstrom resolution. And as I say, that's with no movie frame alignment, so I would hope as soon as we do that, as Abe does that, it should improve quite a lot. And it's just a comparison with the reference structure, which is Abe's 3.2 angstrom. I mean, it looks very nice, and fingers crossed it goes further. So the other thing that we did a while back with Rich Collins and Bob Ford's group was they brought down a 160 kD membrane protein that we collected with standard TEM, and so at the end of the session we thought it would be a good opportunity to have a look at something with the faceplate. So this is a faceplate image, so it's a pretty small membrane protein. I mean, you've obviously got some ice contamination on there, so these bigger blobs, but you can also see that there's a lot of particles in the background. And again, you can see the diffuse water ring and this really, really strong maxima close to the origin, which is why you could see the particles. So the other thing, another kind of commissioning test we did is for tomography. So the nice thing is with the tomography software, the conditioning of the faceplate is actually built into that. So this can be run automatically, and we've had our first external user doing faceplate tomography over the session. So this is a 2-electron image, so the 0-degree image. You can see the contrast is amazing. So then you obviously collect the tilt series, reconstruct. So this is just a movie, and the idea behind this would be to pick the spike proteins on the outside of the virus and do some kind of subtomogram averaging, which again is being done in Dane's group. But it's just I mean, this has not been filtered, so this is just as it comes out of, and the detail is pretty amazing. So then another example so this is more recent. So this is done with Natasha, who's sitting in the audience. So she sent me the movie, so thank you very much. So again, this was 1.5 electrons, a total dose of about 50 electrons. So you see on the left-hand side. So these are vesicles with a pore-forming toxin that's been kind of locked into a pre-pore conformation. Wow, that's fast. Run that again. So again, this is with the faceplate, and so hopefully that's going to play. Okay, fair enough. Try that again. So this is obviously the reconstructed volume, so you can see the bilayer very nicely. You can see the pores on the outside. So that's this guy here, and they're all over. So, I mean, if you've looked at these things, I mean, you can already see that just from the movie frame, you can see the shape of the monomer. It's pretty nice. I think the end goal for us would also be able to offer kind of, so what I've shown you with the tomography at the moment is kind of more molecular tomography, where you're looking at smaller objects and then you're going to do subtomogram averaging to try and get a higher resolution structure. But it would be really nice to combine the faceplate with looking at whole cells, but obviously cells are too thick. So one of the other things that we have, which is I think Corey's going to be primarily looking after, is we also have a SIOS Fib SEM dual beam. So the idea being is obviously your cells are too thick to get the electron beam through, so you're going to use a gallium beam to thin out a wedge of those cells so that you can actually get your electron beam through and collect some tomograms from. So this is one of the first lamellae that came out of the fib. You can see it's kind of a little ice contaminated, but you can see some nice details. So from a projection image here, you can see the kind of the nuclear envelope. You can see some organelles, so nucleus, but you can also see there's a bit of ice damage. So a more recent picture, so again this is from collaboration between Radu and Kai's group. So Kai's group are trying to push the SIOS, they're trying to work out kind of workflows that we can use so we can start offering this to users. And so this is, they've had a difficult time to get the lamellae that you cut in the FIB-SEM, to get them out of the SEM and into the TEM still in a vitreous state. So this is one of the ones that they did have. So these are SF9 cells, I believe, and it's just, you know, you can see that from this montage that Mike did, you can see basically these bizarre ER structures and organelles, nucleus. So hopefully we'll soon be able to combine the FIB-SEM with the faceplate so we can collect some very nice tomograms. So obviously the people in here, so it's Alistair and me, and Corey's just joined us. There's a lot of help from the local staff on site, so Jean, Alan, Nick, who's actually not here anymore, but yeah. You can see Alan, so this is the guy who installed our cryos and is a top engineer, and obviously Sonia, who's been a massive help to us, and obviously Felix and Sasha are also part of the FEI app support team, and then we've got Liam, who's the Gatan engineer, who's currently on site installing our GIF and K2, and obviously the management committee, and you for your attention. Ooh.
Thank you, Dan, for a lovely overview. So, got time for some questions for Dan?
Excellent.
I'll ask you a question. So, is there any
Tom.
circumstances when you wouldn't use a faceplate?
At the moment, if you're working on a large protein, I don't think there's much. You can get to sub 3 angstroms. I think. So most of the time, no. I think maybe if the implementation of the faceplate within the software improves and the reliability of certain aspects, then it might be worth using it. And I think if you really want to push the resolution, you probably want a CS correcting machine with the faceplate. I think is probably going to be the recipe. But yeah.
Martin?
Can I just make one comment?
Oh, sorry. Can you do it?
A lot of people here, and Dan mentioned this new Horizon 2020 project, which is INEX, which is doing a lot of things.
Yep.
But one thing it's doing is providing access for the whole of Europe to a number of sites providing high-end microscopes. Is everyone familiar with INEX?
So
Okay,
we have flyers by our
Okay,
Microscope, so any users that come generally get told about it.
Just for the streaming.
Sorry. So, just to... Which one's working?
Mine, mine.
Sorry. Sorry. So INEX is basically an EU a Horizon 2020 project which just started in September. We started providing access to high-end microscopes around the end of last year. eBIC is one of the centres that will provide access, but also Nissen in Leiden and in Brno in the Czech Republic, and also Heidelberg at EMBL. So you as UK users or European users can apply for peer-reviewed access to all these facilities, and they all have these wonderful new microscopes. Madrid is also involved with helping with sample preparation. We will be organising courses and training, and there's also software development and all that type of stuff funded within it. So if you want to find out more about that, we'll try and get some flyers around at coffee so you can see. But the website is just www.inex-eu.org. So go there, sign up, and it's another good thing for being part of Europe. Okay? Thanks.
Okay, here's the question.
Have you given any groups more than 40 hours, continuous hours on the Cryo?
Sorry, I didn't hear the start of your question.
So have you given any groups more than 40 hours continuous time on the Cryo?
So most of our sessions are 48 hours, but if you get the Friday session and we're not doing a cryo cycle on Sunday, then you can get 72 hours. But we guarantee 48 as such. Or we do our best to anyway.
Yep.
Hi, Dan.
Yeah.
Could you give us some detail about this, making the faceplate, conditioning the faceplate? So how stable is it? What degradation do you see over the course of your approximately an hour? And how long does it take you to redo it?
So the way that we're doing it is it's pretty standard. So you put the faceplate in, or you shift to a new position on the faceplate, because all the faceplate is a slot grid with a carbon film over the top. And so they have, I think, 90 positions per slot. Then I think you have 6 of them in the machine. So you move to a new slot, you wait for about 5 minutes for the film to stop drifting. Then you put your beam on it in your illumination conditions that you're going to collect in. So that is, you really need to make sure about that, and you need to make sure that your beam is focused on the faceplate at the back focal, so it's hitting that. Then you would generally, I think in the manuals it'll say 50 nanocoulombs. They'll tell you, I think we've had various values for that. At the minute we're kind of sticking to the 50 nanocoulombs because the phase shift that you see comes on pretty quickly. So we tend to kind of wait till we see the initial onset, and then we'll see that the tone rings. So if you defocus a little bit, you'll see that the tone rings start to shrink. So your first zero will get close to the origin as the phase shift kicks in. And then once the phase shift, you kind of reach a point where it's stable, and then it should plateau for a while. So your first zero won't move, and then we say, okay, that's the faceplate activated. That's generally, for us, about 8 to 10 minutes on the K2 at the doses we're using for counting. Then with the degradation, we're kind of primarily going on what was done in the recent paper, the Danev paper. So they were suggesting around an hour. We have run sessions longer than that, and you see, it's a bit strange because you still see phase shift, so you see a lot of phase contrast, but the images just look kind of really, really smooth and blurred. There's just something a little odd about them. So that's why we're sticking, and I think that's the idea behind that in their paper anyway, is that you get to this kind of settled phase shift, but then you go further, so you're going over that. But I mean, it could also be that when you're focusing, you're generating satellite volta potentials on your plate, and they could also be affecting. So maybe an hour is the time that there's not enough power in those to affect your central. I'm not really sure. I don't know if anybody is actually. But.
Hi, I was chatting to one of the users who's doing tomography on the faceplate, who just popped in to see one of the lectures yesterday, and they were saying you were having some problems at negative tilt with the faceplate using tomography, and I was just wondering if you knew anything about that, and I was sort of curious about it.
We're not necessarily having problems with the negative tilt. It's
right.
More the volta potential on the faceplate is it's a very, very small area, and you have to be able to hit that absolutely perfectly to get your phase shift. So that requires your optics to be super stable, and so what we're trying to do at the moment is find a recipe within the software that we're using to make that happen as often as possible. And so one of the things that, we've kind of got a recipe that will give us nice phase contrast for the first. So the way that Tomo works is it's collecting the first half of the tilt series, so it goes from 0 to minus whatever you set, and then it'll come back to 0, realign, and then do the positive half. And so at the minute we're in the situation where we can get consistent phase contrast for the first half of the tilt series pretty much most of the time. Then when it goes back to that 0 position, we have some issues. It's kind of 50% whether you'll get a consistent contrast all the way through, so the second half will match the first, or the second half will have a slightly lower.
So can you not just start the tilt series at, not at the centre and then?
Yeah, the problem is that we want to run this automatically, so we want to collect overnight. So at the minute we don't have the provision in the software to do that. I mean, I'm sure that will happen. It's just I think it's kind of new technology, so people are still, including everybody's still trying to work out how to get the best out of the faceplate and how to use it in such a way that gives you the best data. So, yeah.
Okay, I think in the interest of time, we should thank Dan once again and move on to our next speaker.